we were using actual apoptotic cells, not the TMP glycol. So that was a major difference. And I also cite McGawhall's paper where they look at, well, they also deplete MZMs with CL, and they look at the effect on RPMs. And they found that the RPMs can take up that antigen and send it to the PALS, the PALS area, which is T cell rich. But we wanted to see if they can also take it to the germinal center, which is B cell rich. So I guess I'll just briefly go through how we took the thymocytes, we induced apoptosis with the dexamethasone, labeled it with CFSC, then inject those fluorescently labeled apoptotic thymocytes into other mice, and then 5 minutes to an hour later sacrifice them, take out their spleens, and see and look at those 3 cell populations, the marginal zone B cells, the precursor B cells, and the RPMs, and to see, first of all, do they take up more CFSC labeled apoptotic cells with CL treatment, and if so, does that amount increase with time, and also do they enter the germinal center at a, I guess, higher rate or so. So.
You tell people what CL does. So what's CL?
Oh, right. So I'll talk about how basically it can be used to specifically deplete MZMs if it's used at the right concentration. So we inject the mice with CL. It goes to their spleen, and since these MZMs, I mean, they're so phagocytic, they love to eat stuff up. So the CL will basically be taken into the macrophage, and after, I guess, that lipid coating gets uncovered, then there's some sort of some sort of toxin in there that kills the MZMs, basically.
The chondronate. Chondronate is the toxin.
So
The lady said it. To Sarah, she says it interferes with the ATP in the mitochondria respiratory, so it interferes with energy metabolism of the macrophage.
So here's just the table of the different conditions. Indicates if the mice were treated with CL or not, if they were given apoptotic cells or not, and how many minutes after AC injection they were sacrificed. So there doesn't seem to be a major impact on the follicular B cells. If you look at mice that were given CL, they were given CL but no AC, the I guess efficiency of CFSC labeled cells uptake doesn't really change much as you go through the tested time points. But for MZBs and MZPs, if you
Now, one thing, when you say MZBs and MZPs, we should have put, anyway, that's MZB cells. Because most people don't know what they meant.
Yeah, sorry.
And MZP B cells. I remember I wanted to make that change.
I did label those. It's kind of hard to see.
Right.
I did identify those. So yeah, MZB cells and marginal zone precursor B cells. So they do display an increased ability to take up these labeled thymocytes with CL treatment, and they do seem to take up more of these labeled cells as time goes on. And this is just fluorescent images, basically visualizing the same thing that's going on here.
And they may take up, you might point out that they may directly or indirectly take it up. In other words, they pick it up from, from the marginal macrophages. That's a possibility. Because they probably don't just pick it up because they're an apoptotic cell that's too small, or, the little picture. But
those lines treated with CL, so we won't just.
oh, they can't. Okay, CL treated. Well, yeah, that's true. So they don't pick it up in marginal macrophage. So how do they get that material in there? I mean, they probably don't pick it up themselves because they don't have sign R1 mark. Or no, I guess it. Well, what do you think?
I don't know.
How does that stuff get into B cell?
Those cells are already dead, so it's just fragments of it.
And you think they directly get taken up by the MZPs?
Because they got those sugar, just like so they have CD1B, the pick up with the antigen. They have Reynolds receptor, they can pick up sugar antigen. So the marginal zone B cell and MZP, they do have the potential to directly capture the antigen.
All right, but so they know MZMs, I guess. So that's how they point out. So somehow they either get some of the fragments or debris get. They may be, but some of it you believe is direct, because right. Okay.
It's under the condition of MZM are depleted. That's when they capture it.
Right.
Here, the 15-minute time point. It's the one treated with CL that they have more uptake on body.
All right. So what's the next one show?
And this is basically the same thing up here, but looking at RPMs. And the same general pattern with CL treatment, the RPMs, or a greater percentage of RPMs are CFSC positive, and that percentage does increase with time up until the 30-minute time point. That's where it peaks, and then it decreases at 60 minutes, as opposed to these B cell populations where it increases throughout.
So the RPMs could do it, right? They're not depleted, right, Holly?
The part get depleted, but still you can see over 50% still there.
Yeah. So they make. I mean, you show they capture it. So maybe they, you might say, just for simplicity, just say. I mean, for now, until we figure it out, that the RPMs do the capturing, and then in part, and then the B cells may do it directly, MZPs, because they have sign R1 and Marco, and between the two of them, you load up the B cells, the MZP B cells. I mean, it looks like actually the RPMs are loaded up, in this one figure. The RPMs are very loaded.
It looks like they're more internalized in the RPMs than
Right.
in the B cells.
And we're going to get into that, Holly. Don't worry. Holly's worried we're going to forget that. That he thinks the RPMs go inside. Well, they do go inside. So it may be the MZPs and maybe the RPMs, and we promised we'd get back to the RPMs, right?
That's when you finish
After you finish the other three, after they finish the other 12 papers, step by step. Okay, what's that next part?
Okay, these are confocal images, and what we're looking at here are marginal zone B cells and marginal zone precursor cells, and seeing if they can enter the follicle or the germinal center. And so this is with CL treatment, but no ACs. And I guess what's interesting are these two. So no CL treatment here, so intact, or I guess more intact MZMs than in the CL treatment. And they both have ACs, but the ACs here seem to be cleared more than in the ACs here. And presumably because the MZ barrier is more intact in this mouse right here. So that could.
It'd be good to have a Marco staining because to show they are the opposite. Marco would be very high. Right, it's over there. There's the next one. So you might point up that when you point to that, because all you do is see the presence or absence of AC, but the no CL on the other hand up here will have Marco, the same one. So you go back and forth between.
I had a question. This one has CL, but it looks like there's still quite a bit of these Marco positive MZMs.
No, those are PDCs, I think, aren't they? The green ones.
Aren't the MZMs Marco positive?
Oh, okay.
No things Marco positive.
Okay, Holly, what do you think?
That's, I tell you, you guys, if you do short-term CL treatment, you do see the depletion of Cy1 positive macrophages, but I still see some left of Marco positive macrophages. I don't know why, but that's just what I observed. But if you give long-term treatment, then all those cells are gone. And I think I discussed with Chen before.
So
The Marco, yeah,
with
it's not
AC
toil.
So with AC only then they're completely gone.
Yeah, it's different when you do.
Did you choose the right one, the CL, no AC, you still see lots of Marco? That's not, you do see reduction of Marco positive for sure. That's for sure. So I don't think you choose typical one.
Yeah, it looks like you're right. There's too much Marco.
You definitely see the reduction for sure, and even the flow, you do see reduction. But the Marco positive cells, it's not totally gone, but the cell won't get disappeared first. You won't see any cell won't positive after 4 hours, but you still see a little bit left of those Marco positive rings. I don't know why, but that's faced with Yuying's paper. Her first paper showing Yuying from Carter's group. She showed that in CD19 knockout mice don't have much-zone B cells. The first paper she published, she just say, okay, that in the CD19 knockout you don't have any cell positive much-zone macrophages, but same paper she published kind of surprising, showing that there's Marco positive macrophages in the area. I don't understand why, but just most people just say those two antibody, distinguish the same population in the spring, but it seems not exactly. I don't know, but it's probably something.
Or maybe for simplicity you can paste. I know it's late, but cover that up, or any rate, or maybe just realize you're right. That's a question. Maybe look at the other one and say, well, you picked the wrong. You might say you pick the one that has little high, but in general they're not high. It is a CL plus no AC one. It doesn't look exactly like a ring anyway, though. It looks kind of scattered, and it looks deteriorated. You can almost see that it doesn't look as solid. There's sketchy green cells versus this one over here with no CL is a solid green, more laminar or whatever. Okay. More work to be done.
Yeah, here we're looking, and you can see these RPMs here in red, getting into the germinal center. And again, if you compare these two right here, this guy has intact, or at least more intact MZMs than this guy right here, and it looks like this guy also has more clearance of the ACs as opposed to this guy. And you also see more RPMs in here as opposed to this mice that was not treated with CL.
Yeah, you might point out, I'm thinking at the bottom, but start with the bottom. Tell them what Marco is, and and CFSE is white, so CD68 is red, CD68 red pulp macrophage is red, so they kind of start with that bottom part because it's kind of hidden down there.
I guess I'm taking it for granted because you all know.
Yeah. So, but when you show them, make sure. In fact, for the other one over here, the same thing.
Mm-hmm.
Start out by saying, now, in this one, you see this is stained.
I'll definitely mention how the yellow here are marginal zone precursor
Right.
B cells, and you can see the MZP cells migrating into the follicle. And that's what the yellow here is showing.
You probably don't even know what's migrating into the chronicle at that mean, right? It's our first year student, right? Oh, no, not necessarily.
These are going to be mostly second years and over.
But they
In
might still,
faculty.
they still don't know, right?
Right, they might still don't know what that means. Why you want to look at the migration, what would that mean?
Okay, so yeah, I can mention, so if they're migrating into the follicle, then they could potentially be delivering self-antigens that were on these apoptotic cells into the terminal center.
I don't even know what germinal center is. It's a possibility.
Okay,
If
that's
somebody's
true.
not,
Michael depending on last video for like Minlo, Minlo, those guys studying biology, study structural biology. They don't know. They don't understand immunology at all.
So that's kind of why I put in this picture right here. Maybe I should just briefly go through the different compartments. I mean, this is showing the red pulp, the marginal zone, and the white pulp. So I kind of had a feeling maybe not everyone at the conference would be familiar with spleen anatomy. So yeah, I can mention how, you know, in the red pulp, that's where a lot of red blood cells go to die, and that's also where bloodborne pathogens can also be filtered in. And I can mention how in the white pulp you have the T cell rich PALs and the B cell rich germinal center, and you know, these bloodborne pathogens, they get in through the spleen, and once those antigens that they can be presented to B cells, and then the terminal center is where B cells kind of, once they've been presented with the antigen, that's where they kind of proliferate and become specific for that antigen. So then I can kind of, I guess, use that to get into, okay, well that's pathogens, but what about self-antigens?
Mm-hmm.
Maybe.
And I was thinking, you know, what I do in the talk is think about the main thing, you know, like in just a word. Like, you know, so you know what you're saying. All right, here's a structure of a normal spleen, and then over here, you know, is what we found in BXC2, showing the abnormal marginal zone precursor trafficking of antigens. So bring up the question marginal zone. I mean, think of that sort of as one thing. And then you say there's a couple ways to actually, you have 4 experiments. So you might say there's 4 basic ways to look at this. First off, if marginal zone is important, or no, then you say something like, so you want to see about marginal zone macrophages, and one way to study how important they are is they can be depleted relatively specifically by claudinate liposome. And there, when you do that, you see MZPs carry more or grab more of the autoantigen, at least the labeled autoantigen. And also the same for the RPMs. So that's by FACS, you know. And they say, well, we want to look at the same thing, so to verify the location, we know MZMs are around the follicle, so you want to do the same thing by confocal, and then you go through that. This shows a picture representation of that. First, you know, if you could get claudinate liposomes, give ACs, you see them stay out longer. But, you know, where do they go? Like, for example, the claudinate liposome plus 60 minutes, you see now they're kind of inside. And that tracks back to this one up here, the 60-minute, the first panel. No, not that one over there, that one I'm pointing to, the results, that one, figure 3. You know, that is MZPs, those are MZPs. And finally, over here with the you know, the claudinate